So welcome to the PhD thesis defense of Shayna Williams-Burris. My name is Art Arnold. I'm Shayna's PhD mentor, and I wanted to say a few words first about Shayna. The room's full. Shayna's friends have come from far and wide, actually, flown in, driven far across town. We're connected on the internet around the world, as far away as France. Okay, so I wanted to, I get a lot of emails, but some of them are really important. Okay, here's the first email that I had from Shayna.
Wow.
Okay. And so she's just asking if she could come talk to me about the possibility of doing a rotation in my lab in the first year of the PhD program. Several things that impressed me about Shayna from the very beginning. One was, so all of our PhD candidates have very high intelligence, so that didn't make her stand out. Maybe relative to the general population, but not among my PhD candidates. But she had this burning desire to be a scientist. She had already, by the time she got to UCLA, made this deep commitment to become a research scientist. And she can tell you when that happened, maybe the moment that happened. But I think it was a large role was played by her undergraduate mentor, Melissa Coleman, who has come across town from the Claremont colleges to be here today, showing the strong bond that they formed. And I think Melissa had a profound positive effect on Shayna. Now, I remember some years ago at a PhD graduation ceremony in the Department of Psychology, that one of the PhD candidates stood up and talked the view from the candidate, having finally reached the end of the PhD thesis. And what she talked about was that the PhD thesis involves a repeated progressive recommitment to the goal. And I think what that is that when you start out on this, you have a long-term goal that you want to get a PhD and have a research career, that you don't always anticipate everything that's going to take. So if you have the view that there's a distant mountain that you wish to climb, you don't always realize how many intermediate hills there are before you get there, and the valleys into which you may descend in between those hills in order to get there. And in Shayna's case, she started out on one PhD project, and the mice weren't just didn't behave the way they were supposed to behave. At least they weren't showing the kinds of effects. They had to be, in our lab, they had to be sex chromosome effects. So she studied pain for a while, but the mice weren't behaving, so she had to can that PhD project and do what she's going to present today. That was only one of several little hills and valleys that she went over to reach today. But she's presented her scientific research at meetings. She's gotten awards. This is at the OSSC meeting of a couple of years ago, getting an award from the president, Eric DeFries, and Secretary Jackie Schwartz. But Shayna's shown great resilience. She's done what ended up being really, kind of bone-crunching hard work to get here. Long, long days and nights, every day of the week, week after week, month after month. And so she's going to present the results of this. But I wanted to mention a special attribute of Shayna's, and that is that she has emerged as a great teacher. Okay, she has taught a seminar to undergraduates by herself. This is the third year, okay, as part of our cluster course. And also in the lab, she's been responsible for bringing more undergraduates into our lab and mentoring them than anybody else in the entire history of my lab, and I've been at UCLA for 40 years. And this is a picture that's in my office that Jesse Martinez, the guy in the middle, gave to me, and he was one of Shayna's mentees, okay? He's now on his way to, he's in graduate school, on his way to a PhD program. And I think one of the reasons why the room is full is that people have made a connection with Shayna over the years, and her, the people that she's trained have been, are devoted to her, and have come today, some of them to see Shayna's finest hour here. Okay, so so on that note, Shayna.
Thank you very much. Now I'm going to tell you about my work, and this is kind of a subset of these experiments, but I just tried to make it a nice story, and then I'll tell you a little bit at the end of what else I didn't show you. But so the title of my talk is Sex, Chromosomes, and Testosterone, a novel mouse model sheds light on the etiology of Klinefelter syndrome. And Klinefelter syndrome is when men are born with the second X chromosome in their testes. So it was actually first described in 1942 by Harry Klinefelter, and he worked in a fertility clinic where he noticed this subset of men had very similar symptoms, and specifically they had small testes, low testosterone, high luteinizing hormone and follicle stimulating hormone, and gynecomastia, which is the development of breast tissue. But then in 1959, it was associated with the karyotype XXY as opposed to XY in typical men by a scientist named Patricia Jacobs, and I found out it was actually the first aneuploidy ever observed in humans or animals. So it was published in Nature in a record time of 5 weeks. Pretty cool. But all of that being so long ago, it's still very poorly understood and not very well studied. And it's prevalent. It occurs in 1 in 600 live male births, but only about 25% of patients are identified. Besides the diagnostic features and the karyotype, it also causes some other health problems. So it increases rates of obesity, diabetes and metabolic syndrome, and then some illnesses that more frequently occur in women like osteoporosis, breast cancer, and lupus. And interestingly to me, as a neuroscientist, it also increases rates of some cognitive problems. So boys with Klinefelter syndrome have higher rates of autism, and they have very high rates of delayed speech. Up to 80% have delayed speech. They also have some social cognition deficits and really high rates of learning disability in school. And then as adults, they also have executive functioning deficits. So their average IQ is not outside the normal range. It's just a few points lower than siblings or peer controls, but they have these specific deficits that are not very well understood. So men with Klinefelter syndrome have low testosterone, like I said, and low testosterone causes some of these symptoms. So a question in Klinefelter syndrome is which is contributing to these symptoms? Is it an increased dose of X genes or low testosterone levels? So there's some evidence that testosterone is not the only story, even though treating with testosterone is the primary treatment for Klinefelter syndrome. But testosterone doesn't remedy all the symptoms, and other low testosterone syndromes don't show the same features of Klinefelter syndrome, and young boys show signs before they hit puberty, and it's thought that it's actually debated in the field whether young boys with Klinefelter syndrome have lower testosterone than controls, or if it starts at puberty when their testes start to have problems. So a few people have asked me actually, how did you start studying Klinefelter syndrome? And the answer is kind of that I was already interested in sex differences, and I was interested in sex chromosomes, and my project working on sex chromosomes looking at a women's health problem wasn't panning out, as I mentioned. But then our lab developed this mouse model, and it was kind of an orphan project, so I started working on it, and I became really excited about it, because this mouse model can allow you to separate the X gene dose from the low testosterone factor, and it was previously impossible to do that. So, just to give you a little bit of background on hormones, this will be big review for the LNE folk, but there's a lot of non-LNE folk here, so there's two ways that hormones can act. There's activational hormone effects and organizational hormone effects. So activational hormone effects are transient effects, so they go away when the hormones are removed, and these effects usually are turned on and off throughout life. So some examples are the effects of the menstrual cycle and lactation, because hormones stimulate lactation, but then you obviously don't lactate for the rest of your life. So on the other hand, organizational hormone effects are permanent effects of hormones that the effects remain even after the hormones are removed. And these usually act during critical periods, which just means times during development when the certain tissues are more sensitive to the stimulus. There's different kinds of critical periods, but in hormones there's, for example, genital development. So testicular secretions are necessary for the development of the penis, but if you lose your testosterone and DHT, you don't lose your penis. So it's permanent effects, and another one is deepening voice in puberty. So the thing with organizational hormone effects is that they're difficult to distinguish from sex chromosome effects, because activational hormone effects, you can just take the hormones away or give hormones anew and you can see what they do. But sex chromosome effects and organizational hormone effects you have so early in development, like neonatal, even prenatal day like 13 is like, so before you can actually very easily manipulate hormones, these effects have long-lasting effects. So in our lab, we use this mouse model called the 4-core genotypes model. It's a review for the LNE folks, because we talk about it all the time, but it's really great. So in this mouse model, the testis-determining gene, SRY, so in mammals, if that gene is present, then the animal develops into a male with testes and a penis, and usually male behavior, and generally is male. And it's usually on the Y chromosome, which is why XY is male and XX is female. But in our mice, there's a spontaneous deletion that occurred on the Y chromosome, and then not me, because this was before my time, but some scientists used inserted SRY as a transgene onto chromosome 3, and in this way, the sex chromosome complement is independent of gonadal sex, and I'll tell you more what that means. So we have these mice, these male mice that are X, Y minus, where Y minus is the Y chromosome lacking SRY, plus SRY on the chromosome 3, and these are what we call our normal 4-core male, even though they're not so normal. And if you cross them with an actually normal XX female, the males make 4 types of sperm. They make X and Y with or without SRY. So depending on which sperm fertilizes the egg, you can get XX males, which don't usually occur, or XY females, which also don't usually occur. And so you can call these XX SRY males, but we just call them XX males, XY females, or XX females, XY males. And just to be clear, for people that aren't used to this kind of nomenclature, when I say male, I mean it's an animal that has testes and a penis, regardless of its sex chromosomes. And if I say female, I mean it has ovaries and a vagina. And so don't be confused by XX males, because they're males that have X chromosomes. Anyway, so if you compare this, we consider this like a 2-by-2 comparison. So if you compare this way, females versus males, you can see hormone effects. But if you compare it this way, XX versus XY, you see effects of sex chromosomes that exist independent of gonadal hormones. So, and then you can also manipulate activational hormones by removing the gonads or giving hormones. So, for the Klinefelter mouse model, it's called the sex chromosome trisomy mouse model. We start with the normal, quote-unquote, 4-core genotypes male, but this time we cross it with the XXY female. And we get the XXY females by breeding those XY females from the previous slide, and a subset of their daughters are XXY. And it's a little bit of a side note is that we can only do this in a specific strain of mice that are called MF1 that are an outbred strain, and that causes some issues with when you're looking at genetics, because inbred strains decrease genetic variability, and we don't have that advantage in this model, but we can't make these mice on the inbred strain because the XY females aren't fertile. So, just a side note, I'll come back to that way later in the talk, so just keep it in the corner of your mind. But in this case, the XXY females make 2 types of eggs. They make X or XY minus, and then the males still make 4 types of sperm. So, doing the math, they can make 8 types of offspring. So they can make XY minus females, like similar to the 4 cores, or they can make XY minus SRY males. So these are XXY males, and that is what Klinefelter males are. So this is the shorthand that we use. They also make, so they make all of the 4-core genotypes offspring, plus the Klinefelter, plus they also make XYY offspring, which I measured all 8 of these groups, sometimes also with gonadal manipulations in several experiments, but today I'm just going to show you these 4 groups, the XYs and XXYs, males and females, because this is what we consider the Klinefelter comparison. So it's comparing, in the males, basically a Klinefelter male to a typical male, where the Klinefelter male has low testosterone just like in humans, but in the females, you can compare, again, XXY versus XY in the absence of the testosterone difference, because there's actually very low testosterone in either genotype. So if you, again, same thing, if you compare this way, you see a sex effect or a hormone effect. But if you compare this way, you see a sex chromosome effect. And a note here, though, is that if you actually look this way and you see a difference in the males, it can either be a sex chromosome effect or a hormone effect because of the low testosterone issue. But in the females, that's, if you see the same sex in females, it's a good sign that it's actually a sex chromosome effect. And as a lab that's more interested in sex chromosome effects, and also just differentiating sex chromosome effects, that's a really important, exciting aspect of this model. And you can also control, again, for activational hormone effects by giving or taking it away, removing the gonads or giving hormones or both. So I kind of broke down my experiments by each hypothesis, and so I'll present you the problem, the hypothesis, the experiment, and then the results and the conclusion for each of my major experiments I did. So the first problem that we worked on in collaboration with Eric Blaine's lab and Karen Reed's lab was to take our first cohort of SCT mice and measure metabolic features, because men with Klinefelter syndrome have increased rates of obesity and diabetes metabolic syndrome, and treating with testosterone doesn't cure this feature. And this picture is the one you find on Wikipedia for a representative Klinefelter male. So they have mostly central obesity which is kind of considered like the bad kind of fat. And we hypothesized that the mice with the XX karyotype will have the same phenotype, and this will be a direct genetic effect, not due to hormones, not solely due to hormones. So the experiment was to measure body weight and body composition and weight of metabolic tissues, and to do that in male and female XY and XXY mice. And we also did XX and XYY, but I'll, again, just keep it a little narrower for this talk. So to do this, we gonadectomized, which means taking out the testes or ovaries in adulthood, and also implanted a testosterone pellet. And then right before gonadectomy, we used NMR, which is nuclear magnetic resonance, which is a non-invasive way to measure body composition, so like percent body fat, percent lean mass. And we treated them with testosterone for 4 weeks. We did some behavior measures in between, and then after 4 weeks, we collected tissue. We NMR'd them one more time, sorry, and collected tissue. And what we found was that the XXYs are, in fact, heavier and have greater percent body fat than the XYs in both males and females. So that was a pretty exciting finding. And we also see this big sex effect where the males are also heavier and have greater percent body fat than the females, which is normal. But I was giving this talk to my lab, and I didn't mention that, and my colleague just said, oh, so you don't care about the sex difference. And she was kind of joking, but I actually kind of don't. I care more about the sex chromosome size. So if you look 4 weeks later, we saw the same effect. So it was maintained for 4 weeks with testosterone treatment, and again, there was this big sex effect. And we also measured fasting blood glucose, which is an imperfect measure for some kind of insulin sensitivity or diabetes phenotype, but we didn't find anything with that, but I still plan to measure the insulin, as I think one of my committee members will suggest to do again. Let's test it again, I haven't done it. And in this case, though, we did find a sex effect, and it's actually a little bit interesting because Art mentioned he's not sure if it's been shown that after treating with testosterone, if the sex effect is still this big, but yeah, it was just another interesting result from that small study. So to conclude that study, the XXYs do have greater body weight and percent body fat than the XYs, and the difference occurs in both males and females, which is the first phenotype that we identified that we can say there's a sex chromosome effect in this model. So in the second experiment, we wanted to expand some understanding of the metabolic phenotype because in the first experiment we only tested after 4 weeks of testosterone treatment. So in this experiment we actually didn't treat with testosterone, but instead we just gonadectomized without treating with hormones, and also left half the animals intact, and we tracked them over about 6 months or 37 weeks. And so we hypothesized that maybe in the GDX animals we would see a greater genetic effect compared to the intact, because a lot of times hormone effects actually kind of counteract sex chromosome effects. And we also hypothesized that the metabolic phenotype would worsen over age, because we see that actually in the forefores. So we measured body weight and body composition using NMR over a longer time period, and then we collected metabolic tissues at the end of the experiment again. And this is the timeline this time. So at this experiment, all the mice were gonadectomized at 75 days, and they were weighed once a week and NMR'd once a month until we euthanized them at the end. And if you look in the intact mice, you see this pattern where, again, the XXYs are heavier than the XYs, and the males are heavier than females. So it's the same as we found in the testosterone-treated animals. But if you look in the, and I have little arrows for you to see that, and this is the male and female difference. So if you look in the GDX, though, the males are significantly different, even though the lines are much closer together. And the female difference is in the same direction, but it's not significant. But the males are still heavier than the females. And this was a little surprising to me, and there's also a big genetic, GDX effect, gonadectomy effect, where the GDX actually weigh more than the intact, even though the heaviest intact group weighs more. As a group, as a whole, the GDX animals weigh more. And then for the percent body fat, the difference was present in the intact in the males, but not in the females, and also the same in GXs in the males, and not, or in XY were different than XY in males, but not in females. And there's a very obvious effect of gonadectomy where the mice gain fat much more if they were gonadectomized. So, in conclusion, the XXY way more have greater percent body fat than XY, but difference seems to be facilitated by gonadal hormones. So either testosterone that we treated with before or innate hormones have to be present to see this sex chromosome effect, which is kind of interesting. Yeah, and I'll talk a little bit more about that at the end, I think. But the next interesting question that we decided to look at was that men with Klinefelter syndrome are taller than siblings or peers, and there's a lot of evidence on both sides that it could be genetic or hormonal, because there are genes on the X chromosome that control for height that seem to be dosage sensitive, and then there's also at least one gene on the Y chromosome that controls for height in humans. It's not clear that the mice have those genes on their sex chromosomes, but there's also the possibility that it's testosterone, because the surge of testosterone during puberty actually closes the growth plates. So low testosterone leads to longer bone growth. So we thought this would be a really interesting question to answer in these mice, where we can separate the two. And men with Klinefelter syndrome also have osteoporosis, and that also could be a genetic effect or a hormone effect, because it's actually it's pretty well understood that testosterone increases bone mass, but we thought there might also be a contribution of genetics. So I basically just told you the hypothesis that the XXYs will have longer femur bones, and that it might be genetic, and then same thing with, they might have bone health problems similar to humans, like an osteoporosis phenotype. So the experiment was to measure bone length, bone mineral density using DEXA, and bone architecture using micro-CT, where this is the same timeline, because it's actually the same mice, we just took the bones at the end. And DEXA is dual emission X-ray, so it, it's a really commonly used tool to measure bone mineral density. And then micro-CT is microcomputed tomography, which you can think of it as a mini CAT scan. So if you go to the hospital and have to get a CAT scan, we're just doing that on teeny tiny mouse bones. So in bone length, we found this interesting result that the gonadectomized males had longer bones than the intact males, and within the intact groups, the XXY males had longer bones than the XY males. And this stair-step pattern is really indicative of a hormone effect, because the lower the testosterone level, the longer the bones. And we always see it in the males, so that's a pretty strong evidence that it's actually a testosterone effect, at least in mice. But again, mice don't have necessarily they don't have the SHOX genes on the X chromosome that control for height, and it's not clear that they have the Y chromosome gene that controls for height either.
Shayna, I have a question, if you could go back
Yeah.
one slide. The XY intact males and the XXY intact males, is there differences in the serum testosterone levels?
We haven't measured it yet. Yeah.
Because your conclusion that it's entirely due to testosterone might not be, the case. So if you had gonadectomized them and put in testosterone implant, you might still see this XY, XXY male difference.
Yeah, that's true, and then it would still also though be hard to tell if it was organizational versus X chromosome effect. So one of the experiments that I proposed at the end is to do that to the females and see if in the presence of testosterone with that genotype, if you see a difference, which is something we haven't done yet. And all of the testosterone samples are somewhat embarrassingly still in the freezer. We just need to we're gonna send them to a core to get them all measured nicely, and that's one of my to-do list things before I leave. But yeah, that's a really good. And it's also really important to know if their testosterone level is different in the first place. So. Okay, so in the for the DEXA, for the bone mineral density, we found that just the intact XY males had greater bone mineral density than all of the other male groups, and they had higher bone mineral density than all the female groups. And this result was also the same if you measured bone mineral density in the whole femur. So this, I think, again points to a testosterone effect because with our understanding of the XXY mice, they should have low testosterone like the the humans, but the gonadectomized mice have even lower testosterone than those. So the only group that has healthy normal male levels of testosterone is the only group that has the higher bone mineral density. So for micro-CT, we measured a lot of things, and we got the same pattern in each of the measures where intact mice had, so this is percent bone volume to tissue volume, and in this measure, higher values mean better, healthier bone, more bone, more dense bone, but lower measures mean less healthy bone. So we saw that intact had greater bone health than GDX, and males had greater bone health than females, which is kind of to be expected, but we didn't see a genotype effect. We didn't see an effect of the Klinefelter, the XXY genotype, either hormonal or genetic. And if you look at inverse measures, so if you look at trabecular separation, you just see the opposite pattern. So in this particular measure, the intact males had greater bone health than the intact females. And there were a lot of other ones, but all of them pointed in the direction that basically the intact males had the better outcomes than any of the other groups. So to conclude that study, the XXY males have longer bones than XY males, and gonadectomized males have even longer bones. XY males have greater bone mineral density than all the other groups, and male mice have better bone health than females, and intact have better bone health than GDX. And I think all of these point to pretty strong effects of hormones and not much evidence of genetic effects. Although there's still experiments to really rule out the possibility of genetic effects needing to be facilitated by hormones like we saw in the metabolic study. So the next problem is that men with Klinefelter syndrome have testicular dysfunction, and it's very poorly understood. So it's actually thought that boys with Klinefelter syndrome, their testes start to degrade at puberty, and what you see, and it's a little hard to see in this picture, but and it's hard to see if you don't know what you're looking at, but these are seminiferous tubules, and this space in between is interstitial space, and these little purple dots are Leydig cells. And if you have a better picture, you can actually see spermatogenesis happening in the seminiferous tubules. But then if you look at Klinefelter testes, the seminiferous tubules are like smashed. They have these like holes, like vacuoles in them, and they have way more Leydig cells just everywhere. And I said that there. And it's not understood why this is true. So we hypothesized that our mice will have the same problem. So we weighed the testes, fixed them and stain them, sliced them, look at them under a microscope. This is the same timeline as before because, again, it's the same mice. We collected testes at GDX for half of the mice, the ones we GDX, and then for the mice we didn't GDX, we collected them at the end of the experiment. It was actually the mice at the end of the experiment that got the histology measured. And the first result is just that there's a huge weight difference in the testes. So the Klinefelter testes are much, much smaller. And on the left side is the human pictures that I just showed you, and on the right side is the SCT mouse testes. And I think it's obvious to somebody who's stared at this a long time, but I think it's actually pretty clear that this looks a lot like this, and these look a lot like these, and this looks a lot like this. And we actually did do some cell counting and stuff, but we're not sure of the numbers, so I'm not going to present them, but just like gross non-scientific analysis, they seem to be pretty similar. So really the conclusion of that is that they have testes that are very similar to humans, and that it would be very interesting to study them further, and that's something that we're looking for kind of collaborators to do. That's something that I am so interested in, but I'm a neuroscientist, so I'm not going to probably follow that up, but I hope that somebody figures it out. It's really interesting. Oh, because another side note to that is that Leydig cells are the testosterone secreting cells of the testes. So the fact that they have so many more Leydig cells and low testosterone is interesting. After all the metabolic and physical measures, our lab wrote a grant to actually study behavior in these mice, and it got funded, which is exciting for me as a neuroscientist studying Klinefelter syndrome. So just to remind you a little bit, men with Klinefelter syndrome have cognitive deficits, especially in adulthood, in executive function. So this is called a Stroop test, and it's a very kind of loose measure of executive function, but what you're supposed to do is first say the color of the font of the word. And at first it might be hard because your brain's kind of trying to say both, like the typed out word and the font color. But after a few lines, you actually get pretty good at it. But then I, as the experimenter, say, okay, now switch it. Read the word. And at first it's actually a little bit hard to do, but then you can do it. But people who have executive functioning deficits, this is a much more difficult task. And that's a very simple task. There are a lot of more difficult tasks that are really sensitive. And those tasks men have been shown to have, Klinefelter men have been shown to have deficits. So another way to think of executive functioning tasks is impulse inhibition. So preventing yourself from saying it the wrong way is an example of impulse inhibition. Cognitive flexibility, emotion regulation, and working memory. So we hypothesized the mice would show functioning deficits similar to those seen in humans. So we measured two behaviors. We measured novel object recognition, which is basically a memory task. It's not a working memory task. So it's not exactly an executive functioning measure. It's more like general cognitive ability. But they had been shown in XXY star mice, which are I'm going to tell you about in a minute, to be different. So that the XXY, the mice that are like the Klinefelter model mice do worse, so we wanted to test that. And then we also did a task that's an executive functioning task that's reversal learning. And this was with Data Yench supervised me closely on this task and provided all the materials and guidance on them. But the XY star model is another model of Klinefelter syndrome that's used in our lab, but we consider it almost not an inferior model, just a model that's better for different things, because you can't tell organizational hormone effects from gene dose effects in this experiment, because we don't have the female controls that are XY and XXY. We only have XY star males and XY star males. And the thing that's weird about these mice is the Y star chromosome has a pseudoautosomal region that causes it to combine with the X chromosome. So the XY star mice are basically XY mice, but they have this weird little region of the chromosome. But the XXY star mice have their second chromosome they have one X chromosome, and their second chromosome is a fusion of the X and the Y. So they only have 2 sex chromosomes, but they have all the genetic material of 3 sex chromosomes, X and Y. So the outline for this experiment was at 75 days we either gonadectomize them or we sham gonadectomize them, and we thought the sham was important in these mice. I think it's important in general, but when you're doing behavior, it can be especially sensitive to things like stress, early life stress, or this would be like midlife stress. And so we sham gonadectomize the intact animals. And then a month later, we tested them for novel object recognition, NORC. And then directly after that, we started reversal learning. So for novel object recognition, you habituate the mice for 3 days. So in a dimly lit room, it's quiet and not stressful, as much as possible for the mice. Because mice are, if you've ever worked with mice, they're pretty stressed out little creatures in general. So we tried to make everything, comfortable and not stressful. And then on the 4th day, and we also got them used to having objects in the box with them, so these little black pipes. And then on the 4th day, we put them in the same box, in the same room and everything, with 2 grape pipes. And they were allowed to investigate them for 5 minutes, and then they were taken out of the box and put back in their home cage for an hour. And then when they go back into the chamber, there was the same gray pipe and then a new white pipe. And so this is the new object, and this is the old object. And a healthy mouse that's not stressed out will investigate the new object more than the old object, because it will recognize that it's seen the old object before. And mice are, when they're not stressed out, supposed to be naturally curious, and they are naturally curious. That behavior is important for a lot of different behavior tasks. But you can basically measure whether the mouse remembers the old pipe and recognizes it, or recognizes the new pipe. So in this test, we found no difference in the Klinefelter mice, the XXY stars versus the XY stars. And this is a little bit surprising, especially in the intact, because another group did this exact experiment with these exact mice and did find an effect. And at first I was worried that I did the task wrong, because this was not the first behavior task I had done, but I was early in my life as a behaviorist, And but I looked at the data, and it turns out all the mice are doing the task at a healthy level, not that they're not doing the task, which would be like what you'd expect if they were just scared, or some other way of not presenting them the objects correctly or something. So we just, in our hands, don't see the same difference. Then I did reversal learning, and reversal learning is a really cool task because you can actually do the exact same thing in humans. So you don't do it because it requires poking your nose into a hole, but you could do it. And so in this task, the mice were first taught to poke their nose in the center hole, and then they get a reinforcer of sweetened condensed milk, which they really like, behind them. And then when they get good at that, you make it so that when they poke the center hole, 2 lights come on either side, and only one side is correct if they poke into it. So they have to learn by trial and error to go either to the left or to the right to get their reinforcer. And then when they get really good at this, so they start going like, poke to the center, poke to the left, get my reinforcer, poke to the center, poke to the left, get my reinforcer. And then you switch it to the other side. So some mice have a very, very hard time with this. And our measure for this is trials to criterion. So they have to get 16 out of 20 correct in a sliding window to consider that they passed. This is how many trials they took to pass. So the highest was like 1,040 or something trials to pass, and the lowest was like 42. So this data was really variable, and it was actually skewed heavily to the right. So the statistics are done on log2 transform data, but I showed you the raw numbers just because it's harder to conceptualize, I think, on the graph, the log2. But we see that the Klinefelter mice, the XXY stars, in the intacts do worse than the XY stars, and we don't see it in the GDX. So that by itself, actually, even though we can't separate organizational hormones from sex chromosome effects, points to an activational effect of testosterone, because if you take away the testosterone, then both groups, the Klinefelter mice actually do slightly better. I don't think that's significant, but these do worse when you take away their testosterone. So that we think might be a activational hormone effect. And I just said that. So I'm going to go on to the next one. So the last thing is that the XXI mouse model doesn't allow for the separation of the hormonal versus genetic effects. So what about the SCT mice? So we did the novel object recognition, and it's the same paradigm, except for we didn't leave any intact in the SCTs, we just gonadectomized all of them. And there's a little misleading pattern here that the XYs do better than the XXYs, but it's not significant or even close. So in our hands, again, we don't see a significant difference in this task. And that's the conclusion. So to summarize everything a little bit, the XXY mice weigh more and have greater percent body fat than XYs, but it seems to be a sex chromosome effect that's facilitated by the presence of gonadal hormones. They have bone phenotypes like Klinefelter men, but that seems to be a organizational hormone effect, or even an activational hormone effect too. XXY testes and the SCTs are similar to human Klinefelter patients' testes. The XXY and XXY star perform normally on the novel object recognition task in my hands, and the XXY star perform worse on executive functioning tasks, reversal learning tasks, but it might be an activational effect because we only see it in the intact mice. So all of these experiments left a lot of interesting questions that I'm not going to answer, but I will pose to others to answer. So I think that my research has kind of shown, or has shown, that using intact or hormone-treated animals in metabolic studies is probably important to see the sex chromosome effect. You can equalize the hormones, as we saw in the testosterone-treated animals, but gonadectomizing only might not be the best approach. Back to Nancy's question, I said treat the female XXY and XY with testosterone for the bone measures, but yeah, the males would also be interesting. And similarly, treating the XY stars and XXY stars with testosterone for reversal learning, I think would be an especially interesting experiment, because if you can show that you can actually, if it is possible to bring the Klinefelter mice to healthy levels by equalizing testosterone, that would be a very, very exciting finding. And if you can't, then that would point to an organizational or sex chromosome effect, and that would also be an interesting finding. So I think that experiment actually is being done by not me, but in the lab, like currently or imminently. And then I actually attempted to do reversal learning on the SCT, but I ran into some issues. But it seems like so I stopped because I had to move on, and my mice were getting old and sick. But Shayn in the lab has started troubleshooting this already, and it might be working soon. So that would be, or I think it's already, I mean, the reverse test is already working, but not in the SCT mice yet, but it is promising. Sorry. Okay. So also, I didn't talk any about this, but ultrasonic vocalization analysis, pup ultrasonic vocalizations, might be a measure for delayed speech. In our mouse model, either mouse model, because it's so prevalent in boys with Klinefelter syndrome, we think it might be significant enough to measure in the mice. And I have all my data collected for that. It's still in the analysis phase, but I think that's another promising avenue. And then the next kind of big advance for the SCT model probably is we're backcrossing it to the C57 block 6J inbred strain. And this is important because it will decrease any variance due to genetic background. It'll make it more comparable with other studies, because the other mouse models of Klinefelter syndrome, the XY star, and then there's another one I didn't talk about, the XXY model, are both on the block 6 strain. And a goal of the lab in general is to make these mice, not these mice only, but like our interesting genetic models commercially available and more widely used. So by putting them on an inbred strain, that would allow us to do that. Because right now the only, or the main source of these mice are our colony. And lastly, I collected metabolic tissues from these animals, and we have data on these animals for metabolic phenotypes. So I think it would be interesting to use those tissues to do the first gene expression studies in the SCT mice. And I propose with my unlimited imaginary budget to do this with RNA-seq. And the reason is because it would allow us to correlate our candidate genes, which are exon activation escapee genes, their expression with the traits, which would already be maybe informative, but if we had the whole genome data, then we could use network analysis to identify genes that are in the same pathways that might be more directly related to the traits. So that's kind of my dream. But yeah, we'll see. And that concludes my talk. I'd like to thank very much my advisor, Art. He's taken very seriously the role of mentor and teacher over the years, and has been very supportive of me and my work. And the technicians that have been in the lab since I was in the lab, who basically ran the whole mouse colony and all breeding and most of the surgeries and a lot of other stuff. My collaborators, which include all of my committee members and Dr. Tetratis, who has the micro CT and trained us how to do that, and also counseled us on how to interpret the results. And then all of the members of their labs, I didn't put everybody because there's so many that I've worked with over the years, but I thank all of them. And then most of all, my undergrads, because, and Haley is in both because she was my undergrad and then she became our tech. And they were in the trenches with me doing these experiments, like late at night, long days, every day, holidays, and just put in tons of work and were very careful and dedicated and curious, and they were just really great. And working with them has been a pleasure as much as like Art was saying it's good. I just like doing it, so it's great. And then my funding, which includes the LME training grant for 4 years, and then the Hyde Fellowship through the IVP department and DYF, and then the Klinefelter R1 that we got, that Art got, and the other PIs too, actually. Yeah, and that's it.
So I just wanted to mention that the floor is open now for general questions from this group, and then the format is that the general audience is excused, and her, Shayna's PhD thesis committee will remain behind and have a private discussion with her, which is the final oral exam. There is going to be a reception party for Shayna in Terasaki 5100, and you're invited to that. It's organized by Matt Burris, her husband, and any questions?
Mm-hmm. Yes.
Oh, okay. So, like, in terms of X chromosome dosage, normal females, and Klinefelter males have the same dosage, right? They both have 1X inactive, and they
Yes.
have 1X active.
That's right.
So then, are you proposing that there's some sort of an interaction between X chromosome genes and Y chromosome genes?
Yes, that's a great question. It's one of my favorite things to talk about are the X inactivation escape genes. Most of them have Y homolog. And so men with Klinefelter syndrome and also XYY men have 3 doses of those genes that aren't the same genes that are very similar. So they're called like XY gene pairs, and those are really prime candidates for genes that are influential in Klinefelter syndrome. Yeah.
Do any of those form like multi-subunit complexes or anything like that?
I'm not sure about that. No. Yeah.
Thank you. Very nice talk.
Thank you. Matt.
Instead of my curiosity, me and Jenny were talking about this afterwards.
Is it possible?
I'm just pulling you in. Just, like, with the training, maybe, what if they know like the milk is much bigger? If you popped up through cocaine or something, would they learn? Is it a preference? I mean, is it a possibility that just the preference of the milk itself is just changing their learning style?
Do you mean in the SCTs because they weren't doing it?
Yeah.
The test? Yeah, that is a possibility. We considered. We did a lot of things to try to get them. We tried because they're all under food restriction usually, so they're hungry. We tried water restriction instead, and then actually after I had to stop my version of this experiment. In the next group, they tried pellets, which are these like banana flavored little treats, and the mice seem to be doing much better with that. So. It's strange because I did a reward response test first where I measured if they like the milk, if they'll drink the milk, and they'll drink like 7 grams of it in 2 hours if you let them just have free access to it. But as soon as you say like, okay, now you have to work for it, it doesn't seem to work as well. Yeah, it's a good question. Cocaine might work. I mean, David would be able to answer that question a lot more seriously than me.
that's one more. Do women with 3 X's have any abnormalities or any similarities to Klinefelter's?
Yes, they do. So they have executive functioning deficits that are similar, and they're taller, and they have some health problems too. I'm not as studied up on that, but they're actually an example that I didn't cite of why we think there's actually a gene dose effect, not just testosterone effect, because they don't have a testosterone difference.
Did XXY Klinefelter men, are they treated with testosterone, and does that help alleviate any of the symptoms?
Yeah, that's the primary treatment. And some doctors even say that they should start it as young boys and continue their entire lives. And Art's actually gone to meetings with patients, and he says that the interesting hot topic among the patients is like, how do you get your testosterone? Because there's like different ways of administering it. So, and they do report feelings of well-being and increased muscle mass and other like positive, signs. Yeah, it's actually a little bit debated whether it helps with like the cognitive features, and it doesn't seem to be heavily influential in the metabolic features. So, yeah.
I just have a social question. Do men with Klinefelter syndrome identify as intersex? Like, do they use that labeling on them?
Generally not, even though they, so actually, Eric Blaine right in front of me is an expert on this, on intersex in general, and so Klinefelter is considered like an intersex condition by like doctors, but, or the scientific side, but men with Klinefelter syndrome identify as men generally, overwhelmingly, and yeah, so they wouldn't like say that they're intersex. Yeah.
So following up on that, what's their major complaint? Are they happy? Are they depressed? Are they,
So
what
they-
does it show up?
They're, so this is a really variable disorder. Like I said, only about 25% are even diagnosed. So 75% of men go around not knowing. They might find out when they try to have children, and then usually what I've been told is that they kind of like put the pieces together of like, oh, and then there was delayed speech way back then, and there was learning disability, and they didn't do it well in school. They have poor socioeconomic outcomes, and then they have those health problems that I mentioned. But by and large, they don't get diagnosed by any specific complaint, besides infertility. That would be probably actually the answer, is infertility, because that's why more men get diagnosed than any other reason. Yeah. Go ahead. Yeah.
Oh, no, I was about to clap. I'm
Oh,
sorry.
you're clapping.
Okay, thanks, Marty.
Oh, no, that's fine. And then we got it on yesterday. And we're just.
I hope people walk very slowly.
Thank you so much. Thank you.
Shayna, before you leave, make sure you get your.
Okay, it's good. Did they stop the? Okay, that's.
Yeah.
Did you stop the camera?
Yes. So we'll do that.
Thank you. Thank you for
I'm gonna send.
Stop, right?
Yeah.